It's a great pleasure to welcome Professor Steve Prescott from Toronto to this talk that is a QLS, CAMBAM, Allen Edwards Pain Center co-sponsored talk. As usual, if there are questions for the online people, you can put it in the chat, and I think Reza will read it out either during the talk or after the talk, okay, both times. So Steve did his bachelor's in medicine and his PhD here at McGill. He then did a postdoc at the Salk Institute with Terence Sejnowski, and then he was briefly at the University of Pittsburgh as a faculty member, and then he moved to Toronto, where he has been for a while. He has worked on action potential initiation, transmission, ion channel excitability, all with and neural coding mechanisms, all with a focus on pain, especially recently. Primarily on pain. Okay, thanks. And over to you, Steve.
Okay, great. Thank you very much for having me. Hopefully online you can hear and see everything. I'll just assume that that's true. Okay, so I want to start off, there's a little bit of a lag here. Okay, there we go. Hopefully this is gonna work. Start off by trying to convince you that what you perceive depends not only on your sensory input, it also depends critically on how your brain processes that input. So if you look at the two squares marked with a star, I think you'll agree that the one on the left looks blue and the one on the right looks yellow. A little bit bleached out here, but hopefully you can see that, especially online. If I take away the surrounding color, however, you see that they're both exactly the same shade of gray. Okay, put the color back. I don't know about you, but I can't make myself see gray in those two squares marked there. So this is an example where you can create color where there isn't none in those gray squares. Here, hopefully you can see, if you're close enough to the screen at least, some movement. So that's an example of a visual illusion where, again, you're perceiving something that's not actually there. Now, we're doing visual illusions because it's hard to do somatosensory illusions as a group, but there are such things. So I'm particularly interested in the thermal grill illusion. This is an example where you feel burning pain in the absence of a noxious heat stimulus. The idea behind the thermal grill is you have warm and cool bars. You touch the warm bar, feels warm. Touch the cool bar, feels cool. But when you interlace them and lay your hand down across them, you get this burning hot sensation. You might have a similar sort of sensation if your hands are sort of numb from the cold and you go wash them in lukewarm water, and you have the sense of, is this searing hot, pain, or what's going on? Why is it feeling this way? But I think this is interesting because the way in which you might trick the nervous system might suggest ways in which the nervous system breaks down, because spontaneous burning pain is a major clinical problem in certain types of pathological pain, known as neuropathic pain. And that's one of those clinical problems that I'm interested in. So I'm not going to talk a lot. Well, so I'll just say this, which is a lot of that processing ultimately boils down to electrical signals. And this idea of animal electricity goes back to the 1780s when Galvani tried to connect different parts of the frog together to get the muscles to twitch. On this idea that this was an intrinsic property of the system, that there's intrinsic excitability. Of course, you can also try to connect your sheep to a frog and a variety of different things. If you want to be more clinically relevant, you can do this with people, the French Revolution, so maybe you have some heads kicking around. And of course, scientific outreach is always really important as well. So it became very popular to try to use electricity to reanimate recently convicted criminals after their execution, as you see here on the left. And actually it was this sort of event that Mary Shelley witnessed in the 1800s and prompted, it sort of inspired Frankenstein, which is of course the birth of science fiction. So we have a long history of trying to stimulate the nervous system with electricity, but it was 100 years later that recording techniques got to the resolution where you could resolve individual action potentials, or spikes. So these short pulses of electricity, which are ultimately the currency by which information is encoded by neurons. And this goes back to the 1920s with Adrian sort of came up with this idea of rate coding, that the rate of action potentials correlates with the strength of stimulation. It's another 30 years before Hodgkin and Huxley really deconstruct that action potential and tell us how these pulses of electricity are being generated by the interaction of different types of sodium and potassium channels. And that leads us to today. So I'm not going to necessarily talk a lot about pain. This is really supposed to be sort of more computational in nature. So I've got this broken down into a couple of different parts. I'm going to start off by considering the basis for neuronal excitability, how are spikes initiated, and what does threshold really mean, sort of beyond the textbook definition. And then I'm going to move on to the consequences of neuronal excitability. And then time permitting, at the end, get into this idea of trying to homeostatically regulate excitability, because I think this is important for understanding neurological disorders, especially chronic ones. So we'll start off with this question of how are spikes initiated, and specifically sort of what's the nature of spike threshold. So what you would have learned in sort of, basic biology is that spikes are initiated when voltage crosses some sort of threshold. Now, what happens at that point? What is the voltage where voltage-gated sodium channels start to activate, and you get a positive feedback loop that gives you this explosive sort of depolarization. You have the upstroke of the spike. That's a very sort of simplistic explanation, which works for some things, but not necessarily for all types of neurons. So here I'm giving you an example of a repetitively spiking neuron where when you inject current into the cell body, as they showed you down here, when you inject current, the neuron starts to spike, and if you were to inject more current, it spikes more, right? So a good example where the firing rate is proportional to the stimulus intensity. But different neurons will do very, very different things. So in this other example here shown on the right, you have an example where the neuron's only responding when you have a change in stimulus intensity, suggesting that there's actually something quite different about why it's spiking, right? And in fact, we might refer to these as integrators or differentiators, where the differentiator is actually sensitive to the rate of change of stimulus intensity. I'm not going to go into that in too much detail, but just to show you here that we can reproduce that in very simple types of neurons, for instance, with Morse-Lacquer type models, which are sort of convenient in the sense that you can really deconstruct this into fast components and slow components, and then do things like phase plane analysis, where you're really plotting the fast variable against the slow variable to try to understand how those interactions are taking place. Now, I'm not going to go into phase plane analysis and null-clines and whatnot, but just to say here that we can take one simple model and switch it from this repetitive spiking pattern to a transient spiking pattern by varying as few as one parameter, where this amounts to adding a subthreshold current or changing, for instance, the voltage sensitivity of that subthreshold current, which for the aficionados is manifested as a shift in the W null-cline, or the gray curve here, which really corresponds to the voltage at which this negative feedback is being activated. Now, what I want to draw your attention to is essentially comparing left and right, is that on the right-hand side here for this transient spiking neuron, you're getting a lot of activation of this low threshold potassium current, this negative feedback at voltages that are around spike threshold, right? So as you start to depolarize, you're trying to activate those voltage-gated sodium channels to invoke this positive feedback process, but you have to do that faster than you activate these outward potassium channels, which are mediating negative feedback. So you have a race between positive and negative feedback, and that gives you this sort of ability to take the derivative, right, is a race, right? So the timing now becomes important. It's not just about reaching spike threshold, you need to reach it fast enough. There's a second criteria that needs to be met for that spike to happen. Now, with that in mind, I was just showing you examples of two different types of neurons, but I'm going to take that information and actually show you that the soma and axon of a sort of generic pyramidal cell actually use different spike initiation mechanisms, why they do that, and that there's a good reason for that. So what I'm showing you here is an example pyramidal cell in the CA1 region of hippocampus. And a typical experiment, like what I showed you before, is that we can inject current into the cell body and then recording from the same pipette, look at the spiking pattern here. So this is a sort of at threshold, the minimum current to get it to spike. But as you turn up the stimulus intensity, you get more and more spikes, right? Now that sort of tells you something about, how the soma or the cell body generates its action potentials, or things like the axon initial segment, which is acting in very close proximity, sort of collaboratively with the soma. But what about the axon, right? So the axon is responsible for taking these spikes and transmitting them to downstream postsynaptic neurons. But does it have the same spike generation mechanisms, or is it different? Now you might say, well, okay, let's just go patch in the axon and we'll do the same experiment to try to characterize this. Problem is axons are very, very small. And they're covered in myelin. So we have a problem where you can't necessarily do exactly the same experiment in the axon. So optogenetics, lots of people have been doing optogenetics for the last 15, 20 years. The idea is that you have these light-gated cation channels that when you shine light on them, you can activate those channels and cause depolarization. Move our light source over into the axon, and we're going to photostimulate there. And what's notable about this is that we can provide a prolonged photostimulus, which is unlike if you were trying to stimulate extracellularly, you can't provide sort of a sustained extracellular stimulus. It just doesn't work that way. you'd have to actually inject current into the cell, and that's what we can't do because we can't patch there, right? But you don't necessarily need to record from where you're stimulating, because those action potentials that you evoke in the photostimulation site will hopefully propagate back to the soma, and you can record the spikes there, right? So the idea is, let's stimulate one space, evoke a spiking pattern, but then see what that spiking pattern looks like based on recording from a different location. Okay, so we did that. We now move the stimulus over under the microscope and provide a small spot of blue light onto the axon. And what you see here is that as we stimulate with the blue light, it's a sustained stimulus, but no matter how strongly we spike, we only ever get one spike at the beginning. And there's a few extra steps here to deal with the channel rhodopsin sort of kinetics and whatnot. But it's a very different spiking pattern from what we see in the soma. But let's do a little control experiment and just move this photostimulus back to the soma. And what you see is when we do exactly the same sort of stimulus in the soma, we get repetitive spiking, suggesting there is, in fact, difference, similar to that repetitive transient spiking dichotomy I showed you before between the soma and the axon. We can do some other sort of control experiments here just to show that indeed, these action potentials, these spikes are propagating back towards the soma. For instance, if we hyperpolarize the soma, we do not prevent these retrograde, these antidromically conducted spikes, and they have a very sharp, or what's called a kinky spike onset, which tells you that you're recording the spikes that have been initiated elsewhere and are propagating back into the soma. When you're recording proximal to the spike initiation site, you see this very smooth voltage trajectory. So we can sort of differentiate, confirm what we think we're seeing based on that. But it tells us that the axon seems to engage a very different spike initiation mechanism than the same cell is using in its soma. So we took all of our data and used that to build a fairly sort of detailed biophysical model that includes the axon, the nodes of Ranvier and the myelination in between, together, attached to the soma to try to sort of reproduce the same experiments and simulations. And what we could do is we knew that the KV1 channels, these low threshold activating potassium channels, so the potassium channels that activate at voltages at or below spike threshold, that by putting those channels in or near the node of Ranvier, that we could make the axon transient spiking like what we've seen experimentally. But from there, you can make the prediction, well, if we then start to back off, if we start to reduce the density of KV1 channels in the axon, now when we photostimulate in simulations, we now get repetitive spiking, sort of oddly shaped spikes, but that's very similar to what we see experimentally, as I show you here, because now we have a prediction. If we apply 4-aminopyridine, which is known to block KV1 channels at the relatively low doses we use, we get this repetitive spiking as you turn up the stimulus intensity, consistent with the simulations. Now, what's also interesting here is that with 4-aminopyridine, which people will use as like an epilepsy model and whatnot, sometimes you also get repetitive spiking, which is resilient to really strong hyperpolarization of the soma. So this is telling you that you get this ectopic spiking originating in the axon. This is actually sort of interesting because when you think about it, there's sort of a problem here in the sense that the axon, at least the nodes of Ranvier, are packed full, really super high density of sodium channels. And it's a very small sort of capacitor, it's a very small surface area. So they're really sort of explosive. They're like, a tinderbox ready to explode, except for the fact that they have this, what we'd like to think of as a safety switch. They have this potassium channels that keeps that in check. But you really do need the high density of sodium channels to pass, to reliably propagate action potentials down the axon. But then you need to make up for that and prevent this sort of runaway, sort of spontaneous spiking from happening, and that's complements of your KV1 channels. Yep.
So you're changing the conductance of the low voltage potassium channel, both in the soma and the axon, or just doing it?
Yes, we did it in different ways, but it by far, and this is actually borne out in the experiments as well, that most of the KV1, at least what we see is that it's in the axon, that the large majority of the effect is in the axon. And that's different, for instance, for KV7 channels where we could sort of switch off, but the KV7 channels seem to be predominantly somatodendritic. But we have them in both, and we did sort of different, like what you wouldn't easily be able to do experimentally, we sort of lesion them separately in different areas just to make sure. And it's really the axon channels that are important here. Good. Okay. So Now, with that model validated, we did a whole bunch of other stuff that I won't get into, but with that, then we can start to explore the model in ways that are difficult to do experimentally. So what I'm showing you here is just, similar to what I showed you before, is that in the axon, these KB1 channels sort of implement what I like to refer to as a high-pass filter, that basically you need to get depolarization. You need to initiate the action potential before you get strong activation of these KB1 channels, right? And again, if you go too slowly, these KB1 channels, their activation can catch up and they can abort the spike, right? So that race is again happening here. Now, we can then probe the model, for instance, by applying noise, just sort of injecting noise now into the model and asking what's the typical waveform that the neuron likes to respond to, right? So taking a spike triggered average, or here a spike triggered covariance, just ask what features does the axon like to respond to with the initiation of a spike. And what you see here, I'll just point it out in black, which is the spike triggered average, is that there's a very sharp, just a couple of milliseconds of depolarization, or a depolarizing current, but it has to be very intense, but for a very short period of time. Now, what's interesting about this is that that's exactly the waveform, or exactly the current profile that a node of Ranvier will receive when an upstream node of Ranvier generates an action potential, right? So you get an action potential, and then there's a huge sort of wave of axial current flowing down the axon to the next node of Ranvier that's followed by this hyperpolarization. This is sort of turned around because it's being represented in space, and I'm trying to line it up with the filter that you see up here. But essentially, what we see is that the axon, its excitability is designed to respond to the sort of waveform that it receives on a normal basis as part of saltatory conduction, right? So it's an example of filter matching, that it looks for that sort of signal and responds to that by reinitiating a spike at the next node, and it's not responding to other things like sustained depolarization, which would cause ectopic spiking. So the filter is matched to the axial current waveform associated with saltatory conduction. Okay, so to wrap up this part, I'd just like to point out that the soma, or the axon initial segment that's operating in close proximity to it, tends to exhibit, at least in pyramidal cells. That's not true of all neurons, but in pyramidal cells, this repetitive spiking, which Hodgkin back in 1948 would have referred to as class 1 excitability. And that's really good for analog to digital transduction, right? So the dendrites cause somatic depolarization, and the AIS, the axon initial segment, is responsible for converting that into a train of action potentials. But the axon has a very different responsibility. Its responsibility is to take that train of spikes and propagate them reliably to the downstream partners. And for that, this transient spiking, this class 3 excitability is really well suited for that, right? You're just responding selectively to a spike at the upstream node, right? So that's good for transmitting digital signals. So different parts of the neuron tends to have their excitability specialized for their specialized function, right? And I think we don't often think of that because everyone records from the soma. That's how we characterize the excitability of that neuron, but subcellularly at different parts, we know that's different. And people have, for instance, done dendritic recordings and show that dendrites are different, but here we also get to have some insight into axon physiology. Okay, so now what I'm going to do is shift gears a little bit and try to address consequences of neuronal excitability with this issue of, do neurons really use the rate or the timing of their action potentials to encode information? So here I'm just showing you an example spike train of what you see, voltage plotted against time. But if each of those spikes is really a stereotypical all-in-one event, then you can simplify this and just represent each of those spikes as ticks. Now we have what we call a raster, and we've highly simplified the problem. We're just now trying to characterize this sort of simplified version of a spike train. But how would you do that? Well, the simplest way of doing that is just to record the number of spikes you get when you apply some sort of stimulus to the system, right? And so we might call that a rate code, sort of in the spirit of Adrian back in the 1920s. You can do that over a long time window, but maybe you recognize here that it seems like within that time window, it starts off fast and maybe slows down. So maybe we want to count these spikes in a narrower time window. That would also be a rate code. But how wide should that window be? Well, we can sort of keep going and sort of some limit. You get to a point where your bins are so narrow that you only have one spike per bin maximum. And most of your bins have no spikes. right? But then when you look at this looks sort of, very binary, right? Patterns of ones and zeros. So, we can refer to this as a temporal code, and maybe you think, well, maybe there's something in the pattern of those ones and zeros that's important rather than the rate or the number of spikes in any one bin. And so there's different ways of sort of thinking about that. But one way of sort of motivating sort of continued, investigation of this is to ask, well, maybe what's important is the nature of the signal, right? So if you have a slow signal, would a rate code be better than for a fast signal? So just consider that if we try to align this, a slow signal, it sort of makes sense to try to characterize that with these wider bins, right? You sort of get a sense of slow fluctuations in the signal by slow fluctuations in the firing rate. Whereas if you have these fast fluctuations, right, assuming a neuron can't fire super fast, right? There's biophysical constraints on how fast a neuron can spike, but maybe these individual spikes can tell you about specific events in the system. So maybe depending on the nature of the signal, you'd want to look at your spike train in different ways. But if you don't have prior knowledge of the signal and you're just given the spike train and those spike trains look exactly the same, how do you know how to start decoding it, right? And the answer is, well, in the real nervous system, or if you're recording sort of multiple neurons, you don't have to be limited to one neuron to do this. Certainly the brain is not using just one neuron to do these sorts of problems. So let's consider the same signal given to multiple neurons where each of those neurons is either a little bit different, or maybe you have some noise that's uncorrelated, independent from one neuron to the next. So with the slow signal, if you did this, you'd probably see a series of spike trains that looks like that, where you have asynchronous spikes that are co-modulated in their rate. So all the rates are going up at the same time, and they're sort of coming down at the same time, but they're not necessarily synchronized, which is unlike what you'd see with a faster signal, right? Where you do have these precisely synchronized spikes. So now that you have access to multiple neurons and are looking at the pattern of spikes across those multiple neurons, you might be able to start sort of deciphering, which signal was occurring, and at the same time sort of thinking about which way you might want to analyze a spike train, or some insights into how a postsynaptic neuron might be trying to decode these spike trains. But the spike synchrony is really the context in which timing has to be considered, I think, because you get people talking about latency codes, and if the nervous system doesn't know where the start time was, those sorts of codes don't, I think, make necessarily a lot of sense. But these codes, this idea of precisely timed spikes or poorly timed spikes actually resonate, with past experimental work, as seen here, for instance, by putting in, a sustained slow sort of DC step versus noisy steps, where you can get this sort of poorly timed asynchronous spiking versus this highly, precisely timed synchronous spiking. In this case, across trials, but you can imagine it across neurons as well. Okay, so what I just showed you here is really that the stimulus kinetics are important, that if you have a very high contrast, sort of abrupt change, you can get precisely timed spikes, which across a set of neurons will translate into synchronized spiking. Whereas if you have a slower change in your stimulus or a lower contrast, that will tend to give rise to imprecise spiking or asynchronous spiking across a set of neurons. But that really sort of depends on the effects of noise, right? You wouldn't necessarily see this if in your model you had, the same neuron responding the same way to every trial. So you have to add noise to see this. But of course, in experiments, the noise is ubiquitous. But I want to try to explain this a little bit, give you a little bit more intuition here. So here we're just using a leaky integrate and fire model, where you're seeing multiple rasters here because there's different noise on each trial. But what we've done is we actually separated the effects of noise and the effects of signal into different sort of parts, so that the effects of signal are now actually being represented in the threshold. So when you see a drop in the threshold, that reflects an increase in the stimulus intensity, right? And the effects of noise are being reflected in these noisy trajectories, right? So as you see is that as you have a slow increase in your stimulus, that causes a slow decrease in the threshold trajectory. And these trajectories, these voltage trajectories, will now crash into that threshold at different points because of the effects of noise, right? So the timing of spikes evoked by a slow signal are readily disrupted by noise. Now, if you have a much faster event in your signal, despite the different noise, all of these trajectories are going to hit at around the same time, right? So you can actually have conditions where the noise is not very disruptive. In fact, what happens is that the noise can prevent you from maybe smashing into that threshold, but it's going to modulate the reliability. The likelihood of spiking is not actually going to modulate the timing of those spikes very much. But that's really contingent on the kinetics of the stimulus that you're dealing with. Okay, now we're going to use that information and ask, well, so what's happening in the nervous system? Is it rate coding? Is it temporal coding? Or whatever the context in which these could actually be combined? And I'm going to tell you that synchrony is key. So what I'm showing you here are rasters now recorded from primary somatosensory cortex in a lightly anesthetized rat where we poked the whisker pad. So it's not actually moving the whisker, which you might be familiar with. Here we're just poking the whisker pad and asking what's happening in these S1 cortical neurons. And what you see is as we increase the intensity of the poke, you might get a sense that the firing rate is increasing. It's really quite noisy. But if we take a kernel, so it's like counting in a wide bin, so it's a low resolution. We're going to calculate a firing rate histogram with relatively low resolution, counting at a wide bin. You can see that indeed the firing rate is going up each time we apply a stimulus, and it goes up more as you apply stronger stimuli, right? Simple enough. But is there anything more embedded sort of in this signal? So if you now took a firing rate histogram and calculated it using a much narrower bin or with a much narrower filter kernel, what you're going to see is that we actually have these very rapid sort of increases in what looks like firing rate at the onset and offset of every event, right? And what that actually corresponds to is synchronized spiking. So all of these neurons, they might only fire, one action potential sort of, milliseconds after the onset of this event. But as a group, that translates into an increase in the firing rate of the population. And so we can go through and say, well, every spike that's part of one of these very narrow peaks in this firing rate histogram, we're going to color it red. We're going to call it synchronized, right? And all the other ones, well, they're not part of those peaks. We're going to call them asynchronous. And what you see, I can't see it on my screen, but that the rate of asynchronous firing tells you something about the stimulus intensity, but the timing of those synchronous spikes, which are very sparse, but they co-occur with the onset and offset of all of these stimuli, they can tell you a lot about the precise timing of changes in the stimulus intensity or these high contrast events or edges, right? So between the two of them, you can tell when something changes as well as how much, right? But these are spikes in the same neurons, but they're different. They're different because of the degree of synchronization with their neighboring neurons, right? So we refer to this as synchrony division multiplexing, where you have two different signals, you're combining it, but you're able to separate them out on the basis of their degree of synchronization, right? So that's where the spike timing precision comes in. Now, we went into a lot more detail. You can check out the paper if you want more detail about trying to reconstruct, the different components of this signal and does this really work? But it's sort of summed up here that different spikes in the same neurons represent different stimulus features. So as the stimulus changes its intensity, the rate of these asynchronous spikes will go up. That's shown here in the blue. But you really have to sort of look with finer, sort of timing to pull out that there are, in fact, these transiently synchronized, these sparse transiently synchronized spikes. They can tell you about the timing of these high contrast events or when the stimulus changes, right? But you can do both of those using the same set of neurons just by looking at the degree of synchronization. Now, this also works for other sort of settings. So here I'm just showing you, what would be sort of appropriate in the context of vibration, for instance, you can also encode the frequency of the vibration or the amplitude of that vibration or the envelope by again, sort of, using the rate and interval or timing of these spikes. And the key here is that you need to skip these events. The neurons can't fire to every event. They can in train, but they need to skip the events irregularly, right? And that's the benefit of noise, is that you can skip these events irregularly, because if you start skipping regularly, right, of 2 to 1 or whatever, you're going to introduce aliasing. But if you can skip irregularly and the degree of skipping is modulated, for instance, by the amplitude, then you can rate code the intensity and still have a lot of information about the timing, because that slow noise is going to modulate the likelihood of spiking, but you're still going to spike at a very precise phase of these sort of pulses that are being received by the neurons, right? Okay, now for skeptics in the room, you might be thinking, well, maybe this synchrony is just an epiphenomenon of, the stimulus kinetics that doesn't really get used somewhere. And so I'm going to change gears a little bit and give you an example of a spinal cord stimulation where we can actually show that synchrony is actually important for sensation. Okay, so what I'm showing you here is the gate control theory, which originated here at McGill in the 1960s with Ron Malzahn. And the idea basically is that touch inhibits pain. You might know that if you smack your elbow, first thing you're going to do is rub your elbow to try to reduce that pain. And the idea behind the circuitry here is that the low threshold input that responds to touch enters the spinal cord and activates inhibitory neurons that are going to inhibit the pain signals being passed to these neurons through high threshold or nociceptive neurons, right? So this inhibition evoked by light touch can help reduce the pain evoked by stronger stimulation, right? That's the basic idea behind gate control. But it extends that electrical activation of touch afferents can also inhibit pain. So it's interesting that within 2 years of the gate control theory being published, there were some clinicians who tried stimulating to try to reduce pain, try to capitalize on this with electrical stimulation. And the idea here is that these low threshold afferents conveniently have collaterals that go up the dorsal columns of the spinal cord, whereas the high threshold afferents do not, right? And so if you were to put a stimulator over top of those dorsal columns, you could stimulate there, try to selectively activate the dorsal columns where the antidromic spike, the idea is that the antidromic spike will come back into the spinal cord, activate these inhibitory neurons and cause some, analgesia. At the same time, you ought to expect, and in fact, this is what they saw, is that spikes will also conduct orthodromically or in the correct direction, right? And they'll go up, into the dorsal column nuclei, thalamus, cortex, and will evoke a buzzing sensation that's known as paresthesia. But the buzzing is very similar to the effect that you might have get with vibration, which would activate those same type of afferents, right? So paresthesia is like this buzzing sensation, and that's sort of associated with, if you're activating these touch sensitive neurons with this sort of pattern, you ought to get sort of a buzzing sensation. And in fact, that's actually quite useful because you will tend to traditionally adjust your spinal cord stimulation so you get paresthesia over the painful area, and then you're stimulating the right neurons. That's where your analgesia is going to be, right? However, it can be dose limiting because that you turn up the intensity to a point where to get enough analgesia, that buzzing might actually itself become very uncomfortable. So it's notable that really only in the last few years, after sort of more than 50 years of spinal cord stimulation, people just tried changing the parameters around, and they discovered that at very high frequencies, so kilohertz frequencies, you can get very good analgesia without paresthesia. And this is really sort of against the dogma that how do you get analgesia if you're not activating these neurons, or sort of the assumption was that you're not activating those neurons. There was some sort of circumstantial evidence suggesting that those neurons, these dorsal column axons, were not being activated by these high frequency signals. So that's where we came in, just to try to figure out, well, what exactly is happening, because there's a lot of science out there that was not entirely clear as to really what the mechanisms for this high frequency stimulation were. A lot of speculation, but not really, I would say, hardcore science. And so what we wanted to do is just sort of back-to-back compare conventional low rate stimulation with high frequency stimulation, doing this in anesthetized rats, applying spinal cord stimulation parameters very similar to what is done clinically, but then recording from these primary afferents that are present in the dorsal root ganglion, right? You don't need to know any other details. And what we saw, so what you're seeing here are rasters. What we saw is that we got very good activation of these neurons, these dorsal root ganglion neurons, with conventional stimulation, which you see in blue. But we also got very strong activation of these neurons with kilohertz frequency stimulation, which is what you see in red. And in fact, if you just quantify the firing rate, so this is just counting spikes, over a very long, a 20-second time bit in this case, you actually get sort of median firing rates that are very similar between the two, right? Which begs the question, well, okay, if we're activating them both at around the same rate, why are we getting paresthesia with the 50 hertz parameters, but no paresthesia with the 1 kilohertz stimulation? And of course, I've already given it away that synchrony is important. So how do we characterize that? So digging deeper into this, what I'm showing you now is we're looking at the rasters. These are all individual neurons. We're looking at those spikes, and we're going to plot the interval between consecutive spikes. That's the interspike interval. We're going to plot that against time. And what you see is that lockstep, all of these data points, are are right on top of one another because all of these neurons are firing every 20 milliseconds, right? So 50 hertz, every 20 milliseconds, every neuron spikes, right? And that's what you see up here in the histogram. There's like basically one huge peak and everything else is pretty small. But then if you take the firing rate histogram, right, and count this in very narrow bins, what you see is a firing histogram that's very spiky, right? So everything, all the neurons spike, and then they're quiescent for the next 19 milliseconds until the next pulse comes around, and they all spike, and then they're quiescent, right? So you get these jumps up and down in the firing rate histogram. We do exactly the same analysis with the asynchronous spiking. What you see is something very different, that you don't have those peaks. But I'll just take you through this a little bit, which is that if we do the same sort of ISI analysis and plot that against time, what you're seeing here is that now at 1 kilohertz, is every millisecond, we're giving it another pulse, right? And actually what you can see is that the spikes are occurring at integer multiples of that 1 millisecond interval, but the neurons are skipping, right? So nothing can, keep firing at 1,000 hertz, right? But maybe they'll fire, sort of every 2 pulses, or 3 or 4 or 5. And actually what they'll do is they'll burst, and I'll get to that in a second. But by doing this, the neurons all tend to get out of phase. So the spikes occur at integer multiples of the 1 millisecond interval, but they will skip different pulses. So at the population level, this amounts to the population becoming desynchronized, right? So I want to give you a sense of how this might occur. So with 50 hertz stimulation, right, 20 milliseconds in between, all the neurons spike. They have a refractory period, right? They can't spike immediately again, but all of them spike. That refractory period happens, but all of those refractory periods have waned. They're all sort of negligible by the time the next pulse arrives. So the next pulse arrives and they all spike again, right? Make sense? Simple. However, at kilohertz frequency, you're now overdriving the system. You're pushing it to fire faster and faster. So the next spike is going to try to happen while the neuron's still refractory, and it's not going to happen, right? But that refractoriness will eventually wane, and once it's waned enough, that neuron is going to spike, but maybe the refractory period in the neighboring neurons a little bit longer, or maybe the noise is a little bit different. And so because of that heterogeneity, or because of the effects of uncorrelated noise, you're going to promptly become desynchronized, right? So we refer to this as overdrive desynchronization. Yes, please.
Quick clarification. Fascinating. But where did everyone come up to 1 kilohertz? I mean, did they try 2 kilohertz, 500? Did they try to draw that noise?
We did. Okay. So you can get into that. But I think actually, and this is commercial, right? So people get very uptight about like what patents are applicable where. People started off, there's a company called Nevro, which was really focused on 10 kilohertz. And I think the thinking was they were actually testing much higher frequencies, because if you go up to 100 kilohertz and things like this, you can actually cause sort of a buzzing, like where everything blocks, if you get into that regime. Of course, that's actually not good from a clinical point of view, because you don't want to stop all sensory function, which is what would happen if you were actually blocking transmission. But I think they thought they might be blocking transmission, so they turned this down, which also has benefits for battery life and stuff like that. And it sort of found a sweet spot where 10 kilohertz seemed like it worked, and it does work clinically. So let's not worry too much about why and just make money off of it, right? And then other companies, sort of recognized where the patents were and thought, well, they didn't patent 1 kilohertz, so let's try that. And it sort of went on from there. What I think is important is what we saw is that at around 300 hertz, you get this transition from synchronous to asynchronous. So we think there's sort of, and it's a fuzzy sort of transition, but we think that's sort of where you get this switching between effects, right? So even in the high 500 hertz works pretty much the same as 1 kilohertz, works pretty much the same as 10 kilohertz, right? So yeah. Good. If there's more questions, please ask. I'm happy to take them. Yes.
Have you considered doing this in a week lab?
It's yes and no. It would be impossible for us to also simultaneously record from the DRG, just because unlike the cortex, which has a nice sort of hard case to like fix everything to.
Asking them to report the paresthesia and then test the two frequencies.
Yeah, I'll get to this because we haven't done it behaviorally, but I'll show you some cortical recordings which we think sort of speak to cortical activation that reflect this. People have, this is sort of under the assumption, and we actually did some human recordings with very similar stimulus parameters where the human subject could report to us exactly what they were feeling. And in some ways, we figured that's sort of better than trying to figure out a way of making the rat do it. So yeah, we sort of address that, but in a very roundabout way, I'd say. Happy to take more questions about it afterwards when we speak. Yeah.
Sorry. So if I get this right, I mean, the main issue here is that the refractory period is causing that differences in the response to the basic neurons at high frequency.
Yeah.
So could you counter that effect by increasing the amplitude of the stimulus to basically reduce the effect of the refractory period?
Yeah, but you're still going to get an absolute refractory period, and at a certain point, yeah, I mean, I think part of what happens here is that
so what I'm trying to say is that you can turn red into blue, provided that the stimulus is not strong enough.
Right. But you don't want to stimulate too strongly. But you actually get, so here, depending on the frequency at which you adjust to, at I think high kilohertz frequencies, you can put the amplitude really, really high. Let's say 10 kilohertz. That's, now 0.1 milliseconds. That's, you can't follow that, right? So you could put the amplitude way up and you'd still skip. As you turn the frequency down, so as you went towards, let's say, 300, 400 hertz, you'd probably now have a much narrower window where you need to not go too strong on your stimulus amplitude. Otherwise, you'd still synchronize. So it's really sort of trading those two things off, right? But from clinicians trying to program this, it is actually sort of helpful to just turn one dial way up, right? Let's turn the frequency way up. And, we got a lot of flexibility with the amplitude dial. We don't have to worry about it too much, right? Yeah.
So from your graph before, it looked like some of the neurons were like bursting in this periodic fashion, right? Maybe not this one, but one before that.
Yeah, you can see, here.
Like in red, it looks like some of them were oscillating.
Okay,
at
that's
different
perfect.
frequencies.
Let me take you to this one here, because now we're actually doing this in a mathematical model. And what happens is that we know from other sort of work that you have a refractory period, and then you actually have a period of supernormal excitability immediately after your refractory period, right? So when we actually, we're basically just drawing random numbers from a distribution whose mean fluctuates according to this post-spike excitability. But because of the after depolarization, you're going to increase the likelihood of spiking at these intermediate intervals. But eventually, if you miss that opportunity, you have to wait a long time until that supernormal, there's a cumulative sort of refractoriness as well. So there's actually 3 different time scales interacting here. So you get this slow adaptation building up, which is competing with this fast supernormal excitability. And when you've built up the slow process enough that it overwhelms the intermediate interval, then you stop, and you have to wait a long time for that slow process to then that adaptation to go away before you can get another burst. So actually the bursting falls out of an interaction between the intermediate and this really slow process, whereas what I've been focusing on is really the fast refractory period. But there are actually different time scales, 3 different time scales, which ultimately come into explaining this. And it's the second and third that are interacting to explain the bursting.
So what causes this ADP then, this after depolarizing potential in these neurons?
Yeah, so basically, and this is sort of old work, but the idea is that after a spike happens, you get charge that goes into onto the myelinated area, and then that charge is coming back off after the spike has moved on, right? So you actually, this is sort of a inherent, call it a design flaw or whatever, but it's just something that happens with, this based on the way nodes are sort of in close proximity to myelinated areas of membrane, that you get this charge buildup that comes off slowly after the spike, right? And really, you got to think of this as like a really fast exponential and a slower exponential, but we're seeing, right, the combination of those two things is what is manifest in this shape that you see there, right? That's just the sum of slow and fast exponentials summed up there. Okay, so when we do this, in fact, you do see the bursting in this very simple model, which is just, as I say, drawing random numbers from a distribution whose position is fluctuating according to the post-spike refractoriness, which is captured in this just cartoon up here. We do exactly so in this model, you're basically drawing random numbers every 1 millisecond, because that's the interstimulus interval. Right? But if we do exactly the same thing, but draw numbers every 20 milliseconds, which is the interval for 50 hertz stimulation, in fact, here, as you'd expect, the refractory period's gone away, and so you get this highly synchronized spiking. There's still irregularity there. These are different. The parameters are slightly different from one model to the next. But again, you see very similar with this very simple model, very similar to what we see experimentally. We can make a biophysical model that's very detailed, get exactly the same thing, but I'm going to skip over that part because what I want to bring you to is just this idea that it would appear that inhibition can be engaged, right? Remember the gate control theory, the whole idea was that the spinal cord stimulation is somehow activating inhibitory neurons. What we're showing you here is that if we measure the Hoffman reflex is a stretch reflex, right? So you sort of slap your knee and you get a stretch reflex. That's entirely mediated at the spinal cord level. What we're showing is that for both 50 hertz and 1 kilohertz spinal cord stimulation, you can significantly attenuate that reflex, which is telling us that we're engaging inhibition at the spinal level. But we can do exactly the same sort of test at the cortical level by looking at the response to electrical nerve stimulation. And you see that we're attenuating the somatosensory evoked potential to the same degree with 50 hertz and 1 kilohertz spinal cord stimulation. So it's suggesting that regardless of the synchrony, remember we had about the same firing rate, and consequent to that firing rate, we think we're engaging inhibition to the same degree, both at the spinal level and the supraspinal levels by these two types of spinal cord stimulation, right? So the analgesia is actually similar, but then it begs the question, well, why is it that these 50 hertz stimulation, those spikes, how are they reaching cortex and causing paresthesia, whereas the asynchronous spikes seem to not reach cortex and not cause paresthesia? That's maybe a simplistic way of seeing it, but there's somehow the asynchronous spikes don't cause you this sensation. They're happening, but you're not sensing them, right? So what I'm showing you here is a very simple sort of network simulation, as simple as they get, where we're actually feeding in spike trains that we've recorded experimentally, and we're giving those spike trains to activate both inhibitory neurons as well as our output neurons, right? But what's important here is that the inhibitory neurons are then producing feed-forward inhibition onto the excitatory output neurons, right? And so you can see in the differently colored rasters here is that, yes, we are activating inhibitory neurons, but those inhibitory neurons actually do a very poor job. They don't seem to block any of the spikes in the excitatory neurons because feed-forward inhibition arrives just a little bit too late to do anything, right? So you're engaging a lot of inhibition, but it's always happening moments too late to block the spikes that were evoked by the same signal as what activated the feed-forward inhibitory neurons, right? Now, if you were to take a randomly timed nerve stimulus anywhere else sort of in this interval here, it would probably hit a lot of inhibition and not propagate very well. But the synchronized spinal cord evoked spikes sort of have privilege. They occur at exactly when the inhibition is weakest and tend to propagate very well because of that. And that's in contrast to what you see when you have this asynchronous spiking, is that you actually activate the inhibitory neurons very well, right? They tend to behave like integrators, are very happy being activated by asynchronous input, and they totally shut down all the spiking in these output neurons. And we've tested that over a variety of different sort of parameter combinations to show that this is pretty robust. So now if we actually try to test this experimentally, what I'm showing you here on the left is just a spectrogram. While we're recording, we're actually doing this simultaneous with the somatosensory evoked potentials. But what we see is that when we turn on the 50 hertz stimulus, we see this increase in power across a broad range of different frequencies, suggesting that you see a little bit of 50 hertz at first, but it actually gives way to activity that we think is intrinsic to the cortex, but it's revealing that somehow we've done something to the cortex. At least that 50 hertz signal has gotten there. When we do exactly the same thing with a 1 kilohertz, there's nothing. There's no change whatsoever before, during, and after, suggesting that the feed-forward inhibition has very effectively blocked this signal, probably in the dorsal column nuclei, maybe also in the thalamus or layer 4 cortex, but that signal's really not gotten very far because the feed-forward inhibition is much more effective in blocking the asynchronous spikes than it is in blocking the synchronous spikes. So takeaway message from this is that the asynchronous spikes are sufficient to engage the inhibition required for analgesia, but synchronous spikes are necessary for that paresthesia, right? So from a clinical perspective, you can have your analgesia without the paresthesia. All you need to do is try to desynchronize your spikes, and frequency, turning up the frequency, you get this overdrive desynchronization is one way of doing it. you can imagine maybe other ways of doing it, but having that sort of theoretical idea for how to capitalize on knowledge of the axon biophysics, can really help you sort of, identify certain strategic ways to try to optimize your spinal cord stimulation parameters, changing the waveform and things like this as well. Yeah.
So quick question. This was also used in a
Yes, it is.
but you're not done this in a
No. So we could do this in a wave rats. Yeah.
I'm just curious because a lot of this is very reminiscent of auditory physiology. And there's a whole story of anesthesia affecting inhibition and things like that.
Yeah, and that's why we did, for instance, what I told you about first, it was actually in sedated animals, just because we weren't set up at the time to do awake behaving. But it is really important that the inhibition is especially sort of messed up at the cortical level. Urethane's actually really good subcortically, that it actually doesn't seem to do much. The reflexes are intact, and so we really like it for a lot of what we're doing subcortically. But I agree, cortically, it would be really nice to do this. But this is again where, and I'm not showing it just for sake of time in this talk, but where we did very similar analysis in humans and asked them what they're feeling. And I'm not telling you about ECAP recordings or compound action potentials, but we did this in both rats, pigs, and humans to correlate ECAPs with the paresthesia intensity, for instance. But what's important there is that the compound action potential, you only see the compound action potential when you have synchronized spiking. So a lot of people clinically had interpreted the absence of these ECAPs, which can actually be recorded from the stimulating electrodes that you're using to apply the SCS. They interpreted absence of those ECAPs as absence of spiking. What absence of those ECAPs really means is absence of synchronized spiking. You can have lots of asynchronous spiking and it's not going to show up, right? The local field potential is not giving you a big signal because of the desynchronization. Right? Okay, good. Moving on to the very last part, which I'm going to try to go through quickly, but I think it's interesting and it's sort of at a high level. So this issue of regulating excitability, I think is important for understanding, sort of thinking about chronic neurological disorders. So neurons, you might think of them just as input-output machines, that you take some sort of synaptic input and are looking at the firing rate and excitability, right? This propensity to generate spikes, that's going to be really important for this input-output relationship, right? And the excitability is dependent on the density of all of these different types of voltage-gated channels, as well as leak channels and stuff like that, that all sort of come into the mix. And I'm just going to represent those parameters here just sort of generically as X, Y, Z. But what I want to make you think about is that how is it that neurons can continue functioning under such diverse conditions, right? So for instance, you might knock out one channel, and yet these neurons can continue to function. But just sort of think of it that we have a couple of dials here, each representing a different voltage gated channel. Let's say we knock out channel X, we're going to turn the dial all the way down to 0, right? And what I'm showing you here is that that perturbation might cause an increase in firing rate, right? And that's what you're seeing here on the right-hand side. But if you give it enough time, the system is set up, it's got sort of error correcting properties. It's set up so that it's going to take that and it's going to compensate, for instance, by increasing the density, increasing the expression of some other channel, right? And as you compensate with the increased expression of that other channel, this firing rate will come back to normal. And I just want to emphasize here that it's sort of the average firing rate, because obviously you have fluctuations in firing rate, but if you want to maintain your dynamic range, you need to sort of maintain your average within the sort of normal bounds of what these neurons like. So it's important to regulate this on a very long time scale. We're talking timescales of days here. But this really comes down to this ability to compensate, comes down to this idea of degeneracy, right? That you can get exactly the same firing rate or the same excitability on the basis of different combinations of X, Y, and Z, and then obviously there's many more channels than that as well, right? And that's because X, Y, and Z all affect the firing rate. Okay, so degeneracy is important in this sort of context. However, we tend as experimentalists to focus on one sort of thing. As an electrophysiologist, you might focus on excitability, but the neurons care about more than just excitability. What about energy, right? So these dials here are also affecting other properties of the cells that are important. So when you perturb one ion channel, you might disrupt the firing rate, but maybe you're also disrupting energy usage, and you might compensate for one by turning up this other dial. But in the process of turning up that other dial, you might actually exacerbate the change in, let's say, energy, right? So maybe the first perturbation was not actually problematic. But the compensation to that, the same compensation that solved one problem exacerbated another problem, right? And if you're a reductionist, you might have overlooked that. But if you're going to sort of put all of these things together, that's what the cell might be concerned with. So how do we sort of, oh, well, first of all, I just want to sort of identify that the issue here is this, in contrast to degeneracy, pleiotropy refers to the idea that any given channel is actually going to affect multiple different properties, right? So this sort of mapping, sort of, in and out, pleiotropy and degeneracy together are quite important. So in the last part here, I'm just going to tell you that multiple channels need to be adjusted and that diversity is key. Because you might have this exacerbation of this problem here, but if you add another dial, adjusting that other dial, right, you get enough dials, you can make that work. You can adjust both firing rate and energy or whatever property is being regulated back to its set point as well. But each dial has to affect the properties in different ways. And what I haven't really emphasized here is that these curves that I'm pointing to now are all a little bit different. It's not the same channel. It's not a redundancy, right? That wouldn't work, right? Each of these dials has to tweak these different properties in subtly different ways. And then it comes down to this issue of, well, you're not really separating, regulation of rate and energy. You're not using different channels to regulate each of those. There's actually sort of overlap, but the ratios with which you need to change those channels is different to regulate rate than for what it is to regulate your energy, right? So you need to define co-adjustment ratios to regulate rate and energy, and a different set of co-adjustment ratios for each of those different properties, right? And it's important here because the number of adjustable parameters needs to exceed the number of regulated properties for this to work, right? So you might think of this as a set of linear equations, right? You need to be underdetermined, right? That's the equivalent of being degenerate. You need to have more parameters than you have system of equations to constrain this, more parameters than you have constraints. And that will give you flexibility or maneuverability to regulate these things. So what I'm showing you here on the left is all of the combinations of two channels are really small. They're GK and GNA that will give you the firing rate. So that's the solution manifold for firing rate shown in red. And there's a different solution manifold shown in green that is the solution for a certain energy efficiency. But what's important is that those two solutions only intersect at a point. So you no longer have a degenerate solution on if you're trying to co-regulate excitability and energy, right? And we can do this for a variety of things, but if you're regulating too many properties, what you see on the right-hand side is that those points don't intersect. So you might not actually have a solution that satisfies the regulation of three different properties if you only have three different dials to do this with, or three different adjustable parameters. But if you increase the number of dials, right, if you can now adjust other properties, then you convert those curves into surfaces, right? And those surfaces now will intersect at curves, or three surfaces will intersect at a point, right? So it's a race between the number of regulated properties and the number of adjustable dials or adjustable ion channels, right? So we often think people will talk about degeneracy, the diversity of ion channels. There's so many ion channels. Why do we have this? I think the point is, well, you're actually trying to regulate a lot of different things that we're not typically thinking about, but that the neuron has to worry about. It's also sort of interesting from an evolutionary point of view to think about how this sort of allows increasing diversity of ion channels allows for new functions to be regulated, right? But you need to sort of generate the ion channel first. Anyway, so I'm going to just wrap up with this, because what I'm showing you in this left graph is just this idea of transient repetitive spiking that we know, for instance, in chronic pain states, you can, and the thinking is even in axons that maybe you convert from transient to repetitive spiking. You can do that by changing potassium channels, but the relative ratio to sodium channels is actually what's really important. So I'm just sort of changing that around a little bit on the right and showing you that there's a set point, right? This is where the system wants to homeostatically regulate. And the system, shown here as a little tiny ball, is being regulated to that set point by the co-adjustment of all of these different ion channels, right? And sort of in an energy landscape point of view, that system is resting at some sort of minimum, right? Now, one way of thinking about pathology, which I think is the way most biologists think about it, is that you might have some disruption in one particular ion channel, and that's forcing your system away from the set point, right? Up out of this energy minimum. If that were true, all you need to do is block that mutant ion channel or whatever, and the system should come back to normal, Now, more often than not, acutely that might work, but chronically it doesn't work, right? And I think the system remains hyperexcitable. It compensates and it messes itself back up. And I think real problem is that under chronic pathological conditions, the set point itself has been shifted, right? The energy landscape's moved, and now you're actually properly regulating the system to the set point. It's really the problem with the set point that's at issue here, right? And trying to block any one of these ion channels isn't going to do it. You really need to think about how you move the set point back. But I just want to, sort of point out here to link it back with what I was just telling you about before, is that maybe this is a perfectly good solution to a different problem. Maybe this is what the neuron or the circuit or whatever needs to do to solve some other problem, but that solution is incompatible with the problem you're looking at. So if you're interested in excitability, maybe this is now hyper-excitable and you're like, why does the system want to do that? But maybe it's solving another problem that's actually more important. I think if you don't think about those other problems, we're not going to really be able to intervene strategically to get this to work. Schreier has a question. Okay. Okay, so this is the summary here. That I told you that the soma and axon use different cycle initiation mechanisms, complements of KV1 channels in the axon, and that's appropriate for the different functions those different parts of the neuron regulate, that they mediate. But also shown you that rate coding and temporal coding are not necessarily mutually exclusive. In fact, you can combine them, right? And there's really sort of at the multi-neuron level and synchronization that allows you to combine them sort of in interesting ways. And hopefully I convinced you that with spinal cord stimulation, that spike synchrony is actually important, that we can actually perceive differently spikes depending on whether they're synchronized. There's a lot of other evidence for that in terms of rat bell cortex and things like this. And in this final part, hopefully this idea that multiple ion channels need to be adjusted in different ways, just get you thinking, right? A lot of what I showed you sort of cartoonish, but I think there's a lot of sort of meat on that that would be interesting to get into. And with that, I'd like to thank my lab as well as the funding, and I'm happy to take any more questions that you have. Thank you.
We have time for a few questions.
Very nice talk, Steve. So my question regards the rate and temporal code being combined.
Yeah.
So if I understand correctly from your talk, or the example you showed us, you were saying that the rate was encoding the stimulus waveform itself with the intensity of that step or the level that you're stepping to, and that the temporal code or synchrony was encoding the derivative, I guess, of that step.
You can think about a contrast or something like that. Yeah.
Yeah. So I was kind of wondering, because there's a big question now about, as you know, rate and temporal codes can be multiplex, but the big question is becoming, is the information that is carried by a temporal code redundant with that carried by the rate code, or are they actually encoding separate features? So I was kind of wondering if you had looked at that in your case, because if I have if I have a waveform, if you show me what the waveform is, I can easily figure out what the derivative is, right? I can easily get that information, just a linear transformation. So I was wondering if it was if you had looked at non-linearities or anything like that.
I don't think we've looked at it the way that you're probably thinking. What I can say is that we looked at this, and I'll back up and say part of our motivation for this was that early on in the sensory system, and this is true at least in the somatosensory system, where you have slow adapting and fast adapting. So you have neurons that tend to separate this out, but then they tend to reconverge in the cortex. And we're wondering if when they reconverge, are you necessarily losing the information that each of them was carrying, or might you actually be able to continue using it, but now just in a shared set of neurons rather than in separate neurons? And that it doesn't, from a design point of view, it doesn't really make sense that the primary afferents are dividing this up, but it seems like that happens very often. So what I can say is that, we've sort of done this trying to reconstruct the different components of the stimulus using the synchronous, like after we've, identified them sort of agnostic to the stimulus, but just based on their degree of synchronization, and that you can reconstruct different components based on using the different types of spikes. And likewise, I think we got into this sort of more in terms of the periodic signal, the amplitude and the phase locking of trying to separately reconstruct the frequency, trying to, discriminate different frequencies based on the interval or the power spectrum of the spike timing, as opposed to the amplitude, which really just sort of, you discount those spikes over a much longer interval. So I'm happy to maybe try to answer that more offline. I think Al had a question. Maybe we should take that one.
Okay, hi, Steve. It's nice to see you after all this time. Can you hear?
Yes.
Okay, great. I have a couple of questions. The first one is, when you have overdrive, that long, slow process you talked about, do you have any insights what that might be? Because we saw the same type of things in cardiac cells when we had overdrive as well.
Yeah, so the really slow, adaptation process, it's less clear exactly what that might be, but I do think about things like even sodium accumulation and like ion concentration changes that might occur on these longer time scales, especially considering these are, relatively fine, small volumes we do know that as you drive things hard enough, you turn on pumps and they're pretty slow. And so I think that might be part of it. But we've not actually dug into this yet, partly because this is actually the first time we've seen this sort of with axons, that this bursting occurs. And the first part, the refractoriness and the depolarizing after potential is sort of well described, but people usually do this with just pairs of spikes. You need to drive it for much longer with many more sort of pulses to see this slow process. And yeah, we want to look into it, but we haven't gotten further than my speculation. But it's interesting if the cardiac stuff might provide some insights there. I should look into it.
Yeah, yeah. I mean, we never really sorted it out completely, but we thought pumps could be involved as well. But in any case, the KV1 channel was that a transient potassium current you added, or a more of a sustained current? The KV1 channel.
Yeah, it's more of a sustained one. And so we are digging into this further to pharmacologically try to figure out what type of KV1 channel, because not some of them are quite transient. We think that it has to activate a little bit slower than sodium channels, still pretty fast, but ought to be maintained, especially if you want to prevent, for instance, this ectopic spiking. Now, what I will point out is that KV7 channels, sort of M-type potassium channels, will also serve this function, but we didn't actually see when we blocked KV7 channels that they did anything. But this might be, different neurons might use these channels differently.
Great. And one last quick question, and then I'm gone. So with the noise, it was really surprising. It was counterintuitive that with noise, you actually could suppress that slow frequency activity. But if you increase the noise, could that then, what I'm saying is that the level of noise that's critical, or?
Yeah, it is the level of noise and the kinetics of the noise. So I think in the Camelliden paper, which is the periodic signals, we really dug into that. And what we saw was that for amplitudes of noise that reproduce the background synaptic activity that you get in vivo, that actually it seems to be optimal at around those amplitudes to give this irregular skipping. Now, what I will point out is that it's important to have that the kinetics of the noise is also important in that you don't want this to be really, really fast, right? Which is to say faster than the periodic signal, because that could disrupt this timing. But if it's a slow noise and it's slow relative to the signal, then you're not disrupting the timing, but you will, for instance, modulate the probability of spiking on any given cycle by virtue of where you are with a noise that's relatively stationary during that cycle. So the kinetics, noise, and signal are also important, as well as the amplitudes. It also all comes together but it seems to work in terms of the kinetics and amplitudes that you'd expect, for instance, in active cortex.
Thanks a million, Steve. Take care. Nice seeing you.
Right. Thanks.
All right. There's a question. There's a question in the chat.
Do you want to read that? Yeah.
Mike Guevara. So
okay.
He says, there was evidence some years ago for cooperativity between sodium channel activation, i.e. channel gating not independent from channel to neighborhood channel. Has this stuff panned out in terms of effects on threshold?
Oh, I think maybe this is Nature paper that was actually sort of, there was an editorial News and Views saying, Hodgkin and Huxley are wrong because of this cooperativity, that you needed cooperativity to get the kink that they were seeing. And actually, I think it's very much, I alluded to this with the optogenetic part, that's very much, I think, an artifact of, if this spike is occurring in the axon and coming back, you will get a kink because of you're not seeing the activation of the sodium channels happening sort of in real time because you're not recording close enough, right? So I think, you know, nature papers have a way of being, you know, exciting, but wrong. And I think David McCormick responded to this, and I think every sort of card-carrying electrophysiologists recognize, you know, stuff done back in the 50s by Eccles, that this was an issue of not recording in proximity to where the spike was being initiated. So I think the answer to that, if I'm understanding the question properly, is an issue of spatial details that were overlooked in this nature paper.
Great.
Yeah, good. Okay.
Great. Thanks, everybody, and thanks, Steve.
Thank you.